Good day, class. We are here at Unit 2 on biomolecules and cells of Chem 13. Some of you requested na pasadahan ko itong Unit 2 because they're having a hard time understanding some of the concepts and they felt that the group discussion or presentation is not enough. So this is just very quick. Before we go to your biomolecules itself and cells itself, we first look at the different elements present in the living systems. Then see how these are organized in the cell, which is, of course, as we know, the basic unit of life, and look into the properties that render these biomolecules fit for living. Finally, we will wrap up with cellular structures and organelles. So we start with what is the most basic type of matter in living things? Of course, that would be an element. And in the most important tool in chemistry, we can see several elements, but we don't see all of these in living things. In fact, there are only a few. So what elements are we made of? We have Chon or chons. This is an easy mnemonic. But you won't be needing this mnemonic since you will remember these elements by heart when we go to the succeeding units. For most, as you can also see in our book, the four elements in the living organism are the first four, or I call it the big four, are CHON: carbon, hydrogen, oxygen, and nitrogen. uh, our body is 63% oxygen and 25.3% hydrogen. So more than three-fourths of our body is composed of hydrogen and oxygen. And actually, one group already asked this in their class. Why is that so? And I know you all got this right. We are mostly made of water, and water is, yes, H2O. And that is why the next unit, our unit 3, is all about water. Then we are also made up of 9.5% carbon. And in unit 1, we learned how important carbon is. That is why your supposed prerequisite course is organic chemistry. It is mostly or dedicated only for studying carbon. Let's review in your unit 3, 1.3. There are two reasons given why carbon is a fit for living things. One is that it readily makes lots of different bonds with other elements found on living components, and another is its versatility, wherein it can bond to other carbon atoms. We are only 1.4% nitrogen, but it is still one of the big four. And overall, this CHON make up 99% of the mass of most cells. How about the 1%? So these are the others. CHON, so we have also your Sp or sulfur and phosphorus, but we can also have your calcium, potassium, sodium, chloride, magnesium, iron, zinc, iodine, selenium, chromium, manganese, molybdenum, and cobalt. So what property renders your CHON atoms appropriate to the chemistry of life? So why CHON? So why these elements? So the short answer is it is their ability to form covalent bonds by electron pair sharing. So for example, we go to our favorite carbon. So how many valence electrons does a carbon have? So this is a review or a much needed review of your organic chemistry. Your carbon has an atomic number of 6. Let me have a pen here. Carbon has atomic number of 6. That means it has 6 protons and 6 electrons. So you will see in this figure, you have 6 protons, positive ones, 4, 5, 6, and 6 electrons. 1, 2, 3, 4, 5, 6. So the inner shell has 2. 2 because the maximum capacity of this inner shell is 2. Hanggang 2 electrons lang siya. So 6 electrons less 2, we have 4 remaining electrons. So our outer orbital or outer shell, we have 1, 2, 3, and 4 valence electrons. We call the outer electrons valence electrons. And to complete the octet, we call it the octet rule, we need 4 additional electrons to complete it. Octet rule meaning 8. It needs 8 more to become stable. So this one can bind to another, can bind to another, can bind to another, can bind to another. That's why it can bond with your hydrogen, 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 and another hydrogen. So 1, 2, 3, 4, 5, 6, 7, 8. That gives you 8, and that's your octet. So let's go to your hydrogen. What's the atomic number of your hydrogen? So hydrogen, the atomic number of hydrogen is 1. That means it has 1 proton and 1 electron. So how many more electrons does your hydrogen need? So I've said a while ago that your inner electron has only a capacity of 2 electrons. So how many more does the hydrogen need? It only needs 1 more electron to make it stable. That's why if you remember this carbon atom a while ago, it binds with 1 hydrogen only because your hydrogen also needs only 1 more additional electron to make it stable. So the compound that we formed a while ago with 1 carbon and 4 hydrogen is actually the simplest compound, which is your methane, CH4. Now we go to your nitrogen and your oxygen. So I'll give you a minute to try and do for nitrogen and oxygen. Your uh, nitrogen's atomic number is 7, and your oxygen's atomic number is 8. So find out how many valence electrons does your nitrogen and oxygen need. Okay. So for nitrogen, you have 2 electrons for your inner shell. So we're left with 5. So what will we add to 5 to make it 8? So that would be 3 more valence electrons. So actually it can make a triple bond because of that. For your oxygen, you can make at most 3, but it can do single bond with nitrogen and another double bond. For your oxygen, on the other hand, 8 minus 2 is 6. So how many to make it 8? We have 2. So your oxygen actually can have at most double bond, but it can also have single bonds. Okay, I hope you got that. Now, Your carbon-carbon bonds are strong enough to be stable, but not that strong that can't break and rearrange. That is important that we can easily rearrange it, break it, and rearrange. So this makes them an excellent building block. And let's look at this figure. This is from your book. This is acetyl coenzyme E, which shows a lot of functional groups in a single biomolecule. So you have here a thioester, amido, amide, hydroxyl, phosphoanhydride, imidazole, amino, and phosphoryl. So this is achieved by carbon to carbon bonding because of the tetrahedral configuration of electron pairs around a singly bonded carbon. So you cannot just see it clearly in a flat um, sheet of paper, but this carbon is bonded to your carbon here and bonded your carbon here is bound to your carbon here. Also same here and same here. So all of it for them. So that's why for your activity 2.1, this is just an exercise for you to check the bonding of your carbon with other of your chon atoms. So carbon, 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 hydrogen, carbon, oxygen, carbon, nitrogen, and see how many bonds can you form when you uh, put them together. So next is we have your hierarchy of life. So now that we know the elements that we have or what we are made of, let us see how these are amazingly organized to make up a cell, which is the basic unit of life. So since school is more relatable to all of you, let us use that as an example later. So here is the hierarchy of life from your atom to your cell. So we start with your atom. We have already your chon, but we choose a carbon only. Carbon. So your carbon, we've already talked about the most important property of this atom. It has four valence electrons. That is very good for bonding. And if we go to the next level, it would be the major precursors. So here, your carbon is bound to two oxygen molecules. One, two oxygen molecules. So this is actually, I think, one of your activities from the previous slide. So now I think you know already how it is bonded to each other. And your CO2, one C, and two oxygen atoms is called your carbon dioxide. It is now more stable and there is sharing of electrons and they are it is covalently bonded. And the carbon dioxide is converted to your metabolites. This is used to form your building blocks. So building blocks of macromolecules. So you will learn when you go to your macromolecules, what are the building blocks of proteins, what are the building blocks of carbohydrates, lipids, and nucleic acids. So macromolecules now are the bigger macro, large molecules for different types are carbohydrates, proteins, lipids, and nucleic acids. Next, if we go to your supramolecular complexes, these are interactions of the macromolecules such as your ribosomes and multi-enzyme complexes. Then we go to your organelles. So this is more familiar to you. These are the tiny organs inside the cell. For example, you have your plasma membrane, your nucleus, you have your mitochondria in the plasma reticulum, etc. So for example, the plasma membrane, which is made up of macromolecules and has the ability to separate the outside and inside of the cell. And now you have your cell, which is, of course, the most familiar to you. And for example, we have a red blood cell, and it has that plasma membrane, but it has also that hemoglobin protein. So this is starting with your building blocks, which is your amino acid. That's na-zoom in lang siya. Then your building blocks of your protein is the amino acid. And then you can find your proteins here sa iyong mga uh, organelles. So, for example, in the school, here is me, your carbon, and let's say I am good at research, and I research with Mom Lily most of the time. I am stable with her, and I share knowledge with her. So that is me of the atom. Major precursor is me and Mom Lily, for example. And let's say we research with people in the public health department. So we have Mom Ann for the public health department, and we also have Mom Vilma for public health department. And the four of us make a good team, and that is your metabolites, forming the research team of public health. Public health research team, which is already all your building blocks. So we are the building blocks. So let's say we have research team also from your MLS department and research team from your RT department. So that forms the whole of this, your past research team. So there are interactions among other teams like the CAS extension team, your faculty admin. So those are supramolecular complexes. And our CAS is a part of CSU Andrews that has a specific function, which is to produce allied health graduates. But CSU Andrews also has CHIM, CTED, CBEA, admin, and other departments more. So I will not go through cells to tissue organ to organ systems. We will stop only at your cells. So for your activity 2.2, this is so easy. It's already answered in your book and in the discussion. You are just arrange the following. So for your 2.3 properties of biomolecules, you will be truly amazed on how we are made in this section where we will look into properties of biomolecules that reflect their fitness to the living condition. In the previous section, in CSU Andrews Campus, a macromolecule is like our CAS research team, which is composed of building blocks of MLS, RT, and PH research teams. So the question is, how can the CAS research macromolecule team do what we do? And how does our macromolecules, proteins, carbohydrates, lipids, and nucleic acids do what they do? What is special about them and what properties do they possess? So we have here in our book four properties that our biomolecules possess, and this is very important. Actually, always a part of your quizzes, even in other units. First is the macromolecules and their building blocks have a sense of direction or directionality. They have uh, end to end. It can be 3 to 5, 5 to 3. So they have directionality. And macromolecules are informational. It's very informational. They have informations in them. And biomolecules have structural complementarity. So in your book, you can even see the lock and key model. They complement each other. And lastly, weak forces maintain biological structure and determine biomolecular interaction. So kanina, we've already stated why it is important that it's bonded weakly so that we can easily rearrange these um, atoms and these molecules. So we go to the next picture, and this is first on our list, biomolecules have a sense or directionality. They don't just bind anyhow to build something. And in this picture, you will see the end-to-end chemical orientation of a single strand of nucleic acid. So in a single strand of a nucleic acid, you will see that there's a sense. You have here the sense. You have here the five' end and a three' end. And nucleic acids actually can only be synthesized in vivo in the five' to three' direction. So it's a bit vague at the moment, but when we go to your nucleic acids, you will understand what this five' to three' direction mean. So in the next units, you will encounter head and tail in the structures of building blocks of biomolecules. So polymerization to form macromolecules occurs by head-tail connection as seen is in this figure. So next picture is a map shows the second important attributes of biomolecules, which is that they are informational. And these are actually the um, amino acids. So when we go to translation of proteins, you will understand that if the combination of these amino acids, no, the combination of your thymine, adenine, and cytosine will give you one amino acid, and this certain amino acid can give you one type of protein. So this is an example that macromolecules are informational. So similar to how the letters can form words when arranged, the sequence of monomeric units has the potential to contain information. So the this figure shows you the third property, which is that biomolecules have a characteristic three-dimensional structure that provides means for molecular recognition through structural complementarity. So the typical analogy is connecting pieces of the puzzle as seen in your text. And this is the other picture in your book two, your lock and key. You have your macromolecule and a specific complementary ligand. So finally, the next picture in the activity shows weak forces. We have here the hydrogen, not the hydrogen ones. So you have here a protein, native double helix. So it needs to be weak, and being weak sometimes is a good thing. Being weak is a good thing in this case since it can continually form and break interactions. So these weak forces restrict organisms to a narrow range of environmental conditions, such as, for example, here your temperature. Sometimes by ionic strength and relative acidity. Actually, if you have your laboratory, you will see and witness this um, effect of weak forces. Hence, this picture on denaturation, the natured protein, it unwinds because of the heat and it unwinds more. So it has separate strands already, but it also have its renaturation phase wherein upon cooling, it will bind again. So in your activity 2.3, it's simply to differentiate hydrogen bonds, van der Waals forces, ionic interactions, and hydrophobic interactions, and give examples of each in biomolecules. So I did not include this much already for the discussion because you have this already in your senior high school. And finally is the organization and structure of cells. You are very familiar on this one. And from your book, just like we believe that we came from a single ancestor, all cells share a common ancestor and fall into one or of three broad categories. Archaea and bacteria are referred to as your prokaryotes, and these are single-celled organisms that lack nuclei and other organelles. On the other hand, your eukarya or you yourself, you are a eukaryote, and eukaryotes have true nuclei and other organelles. So, which one is a prokaryotic cell? Is this A or B? So, prokaryotic cells, this is easy to answer, guys. Prokaryotic cells are much smaller than your eukaryotic cells. Why? Smaller surface area to volume allows nutrients to easily and quickly reach inner parts of the cell. So before we feel superior to our messy sister domains, note that prokaryotes have some advantages. They can live in diverse habitats and grow rapidly, which allows them to evolve quickly and adapt to changing environments. But these only depend on diffusion to obtain and eliminate much of the waste products. That's why they need to be small. And eukaryotic cells are larger and cannot pass nutrients as quickly, and they require specialized organelles to carry out metabolism, provide energy, transport chemicals throughout the cell. So though your prokaryotes and eukaryotes look so much different, they also have some shared characteristics. They perform the same basic functions, and they are surrounded by plasma membrane to control what enters and leaves the cell. So they are filled with cytoplasm. Both of them are filled with cytoplasm. And both of them contain ribosomes to make protein and contain DNA to give the general instructions for cells life. So again, what are the shared characteristics of prokaryotic and eukaryotic cells? So first, they perform the same basic function. They are surrounded by plasma membrane. They are filled with cytoplasm. They contain ribosomes to make proteins, and they contain DNA. So that's fine. Basic functions, plasma membrane, cytoplasm, ribosomes, DNA. So this is the common structural features of bacterial cells. The common structures of your eukaryotic cell, your animal cell specifically, the structures of your plant cell, also another eukaryotic cell structure, and what new cells are found in plant cells or new structures found in plant cells. So there are only three. First is the cell wall. Next is your chloroplast. And then next is your chlorophyll. So cell wall, this provides extra support for the cell and gives it a shape. In other words, if there was no cell wall, then the cell will have no shape. Next is your chloroplast. So chloroplast, these make food for the plant and they are green. Chlorophyll is the very important part of the plant. It's very important in making food for the plant, and this structure takes in sunlight and makes sugar for the plant to eat and become green. So again, what are the three new structures found in plant cells? We have cell wall, chloroplast, and chlorophyll. So actually, I did not already tackle each of your features for your, especially for your eukaryotic cells. That's already part of your activity. And also, I assume already you know a lot of this already from your senior high school and high school itself. So as a summary, we talked about the elements of life. We have the chon, the hierarchy of life from your atom to your cells. The properties of biomolecules, we have four, starting from the sense or directionality, ending with the weak forces. And then we have the organization and structure of your cells. I only highlighted here the shared characteristics and the major differences between your prokaryotic and eukaryotic cells, the animal and the plant cells. And it's already your assignment already to do or look into the different structures of the cells. You know that completely already. Your mitochondria, your Golgi apparatus, endoplasmic reticulum, yes, all those. So that's it. Uh, Thank you for watching.